Hello friends, I am Dr. Sunita Pundale and I have been teaching microbiology and molecular biology to undergraduate and postgraduate students for the past 20 years. After having talked about all the enzymes and accessory proteins required for prokaryotic DNA replication in our previous video lectures, we are now in a position to talk about the actual process of prokaryotic DNA replication. DNA replication takes place in three stages: initiation, elongation, and termination. In today's module, we will be talking about initiation and elongation, and we will discuss termination in the next lecture. Initiation of DNA replication means recruitment of the entire replication machinery at the origin of replication, followed by primer synthesis. DNA replication in E. coli begins at a specific site called oriC. An origin of replication is a DNA site at which DNA replication begins and which is essential for proper replication to occur. Some features of the origins are conserved in bacteria. Like mentioned just now, the replication starts at a particular point called the origin of replication or oriC. The minimal origin in E. coli is 248 base pair long and is 80 rich. There are three 13 base pair sequences which are also called the 13-mer sequences, and these are 80 rich regions. Because 80 pairs are held together by only 2 hydrogen bonds compared to the 3 in the GC base, this region is easier to unwind or undergo local melting. This makes it ideal for opening up the DNA double helix and forming the replication bubble where new strands can begin to form. The origin also includes 4 9 base pair sequences called the 9-mers with the consensus sequence TT-AT-CC-ACA as can be seen on your screen. These 9-mer regions are binding sites for the initiator proteins, DNA A proteins. These 9-mers are therefore sometimes called the DNA A boxes. This is the sequence of oriC. You can see the 13-mer regions and the 9-mer regions. 2 of these 9-mer regions are in one orientation, starting with TT-AT-CC-ACA in one direction, and 2 are in the opposite direction with the same sequence starting in another direction. Let us now see the details of priming at oriC. This is the region of oriC where initiation is going to take place. The first step is the formation of the initial complex. First, DNA A proteins bind ATP and forms a multimer. Along with the HU protein, the DNA A ATP complex binds to the DNA encompassing the 4 9-mer regions. In all, this complex covers about 200 base pairs. HU protein probably induces the bend in the DNA which is seen in the picture on your screen. This bending presumably destabilizes the DNA double helix and facilitates melting of the DNA to form the open complex. The second step is formation of the open complex. The binding of DNA A along with the bending induced by HU protein apparently destabilizes the adjacent 13-mer repeats and causes local DNA melting there. This allows the binding of pre-priming complex DNA B and DNA C. The next step is the formation of the pre-priming complex. DNA C binds to the DNA B protein and helps to deliver it to the DNA complex there. We have seen that the oriC in E. coli contains an AT-rich region and 9-mer regions are called DNA boxes. DNA replication is initiated by the binding of DNA A proteins to the DNA A box sequences or the 9-mer sequences. This binding stimulates the cooperative binding of an additional 20 to 40 DNA A proteins to form a large complex. Other proteins such as HU and IHF also bind. This causes the region to wrap around the DNA A proteins and separates the AT-rich region. Once the DNA A strands are locally separated at oriC, the DNA C protein, that is the helicase loader, brings the DNA B helicase, a hexameric ring-shaped enzyme, to the origin, but in this process, the DNA C is released. The helicase is composed of 6 subunits. It travels along the DNA in the 5' to 3' direction and uses the energy from ATP hydrolysis. The helicase separates the DNA in both directions, creating 2 replication forks, paving for the bidirectional replication. DNA B, that is the helicase, stimulates the binding of the primase, that is DNA G, completing the primosome. Priming can now occur. One primer is made for the leading strand and multiple primers are made for the lagging strand, which is synthesized in the short fragments. Once the primers are laid down, the DNA polymerase 3 holoenzyme is recruited to the replication forks. This multimeric complex forms the replisome, a dynamic protein assembly responsible for synthesizing new DNA strands. The sliding clamps are loaded by the clamp loader to ensure high processivity of the polymerase. Let us now move on and talk about this complicated process of elongation and see the intricate details of elongation in prokaryotic replication. All enzymes and proteins thus form the multi-protein structure that assembles at the bacterial replication fork, forming a replisome to undertake the synthesis of DNA. It contains DNA polymerase and other enzymes and proteins about which we have already talked in detail in the previous lectures. Once the pre-priming complex has assembled, the RNA primers are laid down at the oriC. The elongation phase of DNA replication then begins. This phase is primarily carried out by the DNA polymerase 3 holoenzyme, the principal replicative polymerase in E. coli. It synthesizes both the leading and the lagging strands with remarkable speed and accuracy. The antiparallel structure of the double helix affects replication. DNA polymerases add nucleotides only to the free 3' end of the growing strands. Therefore, a new DNA strand can elongate only in the 5' 3' direction. Along one template strand of DNA, the DNA polymerase synthesizes a leading strand continuously, moving towards the replication fork. To elongate the other new strand, called the lagging strand, DNA polymerase must work in the direction opposite to the direction of the movement of the replication fork. The lagging strand is synthesized as a series of segments called Okazaki fragments. The primers are later removed and gaps which are created are filled by the DNA polymerase 1. The nicks which are left are finally sealed by the DNA ligase. So this process which we saw in short just now, I will be trying to explain it in the coming slides. Replication always proceeds in the 5' 3' direction on both strands, but due to the antiparallel nature of DNA, synthesis occurs differently on the two strands. This is the origin of replication. The primers of the leading strands are laid down. The leading strands are continuously synthesized in the direction of the movement of the replication fork, and the lagging strands are synthesized discontinuously in the form of Okazaki fragments in the direction opposite to the movement of the replication fork. Let us first try to understand the leading strand synthesis. During DNA replication in prokaryotes, the double-stranded parental parental DNA unwinds at the origin of replication to form a replication fork. At each growing fork, a single primer is laid for the leading strand. To this template primer junction, a beta sliding clamp is loaded. The DNA polymerase binds to it and starts synthesizing the leading strand continuously in the 5' 3' direction. Growth of the leading strand proceeds in the same direction as the movement of the growing fork. The core of the DNA polymerase for the leading strand slides over the template and reads it and goes on adding the complementary nucleotides to the growing strand. The lagging strand synthesis is very complicated and it takes place in the form of Okazaki fragments. This is the single-stranded template. The primase binds and synthesizes the first short RNA primer. The DNA polymerase binds to this template primer junction and synthesizes the Okazaki fragment. On completion of the Okazaki fragment synthesis, the polymerase dissociates and binds to the second RNA primer for the Okazaki fragment 2 and completes the synthesis of Okazaki fragment 2. The primer removal takes place followed by gap filling and sealing by helicase. The replication of the lagging strand takes place in the direction opposite to the movement of the replication fork. This is what has to be understood. Thus, DNA polymerase synthesizes the leading strand continuously by sliding over the template. The primase synthesizes short primers at repeated intervals. The DNA polymerase synthesizes the Okazaki fragments in the 5' 3' direction, which I repeat again is opposite to the movement of the replication fork. The DNA pol 1 removes the RNA primers and also fills in the gaps. Finally, ligase seals the nicks between the Okazaki fragments. Thus, fragments 1, 2, 3 and 4 synthesis takes place and lagging strand is synthesized. Thus, if we see the overall direction of replication, we can see that leading strand synthesis takes place continuously, whereas lagging strand synthesis takes place discontinuously in the form of Okazaki fragments. In order to understand this important concept of synthesis of lagging strand and leading strand in opposite directions, we have to answer two very important questions. The first is, because discontinuous synthesis of lagging strand must involve repeated dissociation and reassociation of the core polymerase from the template, this model raises the question, how can discontinuous synthesis of lagging strand possibly keep up with the continuous synthesis of the leading strand? And the second is, how is repeated dissociation and reassociation of the pol3 core from the template compatible with the highly processive nature of DNA replication? After all, the beta clamp is essential for processive replication, but once it clamps onto the DNA, how can the core polymerase dissociate every 1 to 2 kb as it finishes one Okazaki fragment and jumps forward to elongating the next? The answer to the first question seems to be that Pol 3 core making the lagging strand does not really dissociate completely from the template after the completion of each Okazaki fragment synthesis. It remains tethered to it by its association with the core that is making the leading strand. Thus, it can release its grip on the template strand without straying far away from the DNA. This enables it to find the next primer and reassociate with its template within a fraction of a second instead of the many seconds that would be required if it completely left the DNA. Let us revise the structure and the composition of the multi-subunit enzyme DNA polymerase 3 to further understand this complex process. This is the leading core and this is the lagging core. This is the gamma complex, the clamp loader which loads the sliding clamps. The two core polymerases are dimerized by the tau dimers, and that is why these two core polymerases do not have independent existences, but they are part of one big DNA polymerase 3 holoenzyme. As the helicase moves ahead and brings about unwinding of the two strands, the leading core very closely follows it and replicates the leading strand template. At that time, the lagging core is far behind, replicating an Okazaki fragment. As a result, the lagging strand template loops out. The lagging strand is synthesized in the direction opposite to the movement of the replication fork and also to the movement of the DNA polymerase. Hence, the core of the lagging strand does not slide over the template. Rather, the template is pulled through the core, and as it is being pulled, it is read and complementary nucleotides are added. As the template of the lagging strand is pulled through the core, it loops out furthermore. This looping out mechanism is explained by the Trombone model. The coordination between the leading and the lagging strands, the looping out of the template for the lagging strand enables a dimeric DNA polymerase 3 holoenzyme to synthesize both strands because it now appears that both the strands are being synthesized as the complete DNA polymerase 3 holoenzyme is moving ahead. Let us now understand the trombone model. We have seen just now that the helicase separates the two strands. The leading strand is replicated immediately, whereas the lagging strand template loops out and the SSBs come and coat the lagging strand template. The two core polymerases are part of a single protein complex even though they move in opposite directions with respect to each of their templates. Replication of both strands by the two tethered polymerases can be done by having the DNA of the lagging strand loop back on itself. So it has the same orientation of the leading strand template as we have discussed in the previous slide. Both polymerases can then move together as part of a single replicative complex without violating the 5' 3' directionality rule for the synthesis of DNA strand. The lagging strand polymerase is completing the previous Okazaki fragment. Meanwhile, the primase has synthesized an RNA primer on the lagging strand. Once the polymerase assembling the lagging strand reaches the 5' end of the Okazaki fragment made during the previous round, the lagging strand template is released. The loop slides down so that the new primer, which now forms a template primer junction, is ready. The clamp loader loads the clamp to that template primer junction now. The core polymerase now binds to this template primer junction loaded with the beta clamp and starts synthesis of the new Okazaki fragments. And the same sequence of events continue for the synthesis of the lagging strand. Let us have a look at this animation that explains how the replication machinery might exploit the flexibility of DNA to synthesize both the leading and the lagging strands in the same directions by forming a loop on the lagging strand. Having understood this basic looping out model, which is called the trombone model of replication, let us now move on and talk about a model for the cycling of the polymerase 3 complex between the beta clamps on the lagging strand. Polymerase 3 synthesizing the lagging strand must dissociate from one beta clamp as it finishes one Okazaki fragment, and it should go and reassociate with another beta clamp to begin making the second Okazaki fragment as depicted in the figure. But does this dissociation of Pol 3 from its beta clamp actually occur, or does it carry its beta clamp with it? Studies have shown that polymerase dissociates from the beta clamp and binds to another template primer junction already loaded with a beta clamp. Thus, one beta clamp would be required for each Okazaki fragment. Let us look at this animation which is showing the same thing. The E. coli genome is 4.6 MB long and its lagging strand is replicated in Okazaki fragments only 1 to 2 kb long. This means that over 2000 priming events are required on each template so that at least 2000 beta clamps are needed. Because an E. coli cell holds only 300 beta dimers, the supply of beta clamps would be rapidly exhausted unless it is recycled. For recycling, beta clamps need to be unloaded. For unloading, a clamp unloader would be required. Or would the clamp loader act as a clamp unloader? Let us try to understand this. If the clamp is free in the cytoplasm, the gamma complex acts as the clamp loader and loads the beta clamp onto the template primer junction. Once the clamp is loaded, the replication fork proceeds in the 5' 3' direction, and once the Okazaki fragment replication is completed, then the beta clamp is already loaded on to the DNA duplex. At this point now, the clamp loader would act as a clamp unloader and unload the beta clamp, releasing it in the cytoplasm. Thus, beta clamp interacts both with core polymerases and the gamma complex. So let us try and understand this. Beta clamp can interact with both the core polymerase and the gamma complex, that is the clamp loader. It must associate with the core during synthesis of DNA to keep the polymerase onto the template. Then it must dissociate from the template so that it can move to a new site on the DNA where it can interact with another core to make a new Okazaki fragment. This movement to a new DNA site, of course, requires the beta clamp to interact with the clamp loader again. Again, there is a crucial question. How does the cell orchestrate the shifting back and forth of the beta clamp's association with core and with clamp loader? How and when the core and the clamp loader interact with the beta clamp? Let us try and understand this. The core and the clamp loader both contact beta clamp at the same site. It means that beta clamp has the same active binding site for both DNA polymerase and the clamp loader. The beta clamp can bind to the core or the clamp loader, but not both simultaneously. But which one does it prefer? When the beta clamps are free in the solution, beta prefers to bind the clamp loader rather than the core polymerase. This is understood because free beta clamps need to be loaded onto the DNA by the gamma complex before it can interact with the core polymerase. However, that situation should change once the beta clamp is loaded onto the primed DNA template. Once that happens, beta clamps now need to associate with the core polymerase and begin making the DNA. Once the holoenzyme has completed an Okazaki fragment synthesis, it must dissociate from the beta clamp and move on to a new one. Then the original beta clamp must be removed by the clamp loader from the template so that it can participate in the synthesis of another Okazaki fragment. Thus, the gamma complex is both the clamp loader and also the clamp unloader. But what determines when it will load clamps and when would it unload them? We have just now seen that. Thus, when beta clamps are free in the solution and there is a primed template primer junction available, the clamps associate preferentially with the gamma complex, which serves as a clamp loader to bind the beta clamp to the DNA. Once associated with the DNA, the clamp binds preferentially to the core polymerase and sponsors processive synthesis of the Okazaki fragment. When the fragment has been synthesized and only a nick remains, the core loses its affinity for the beta clamp. The clamp reassociates with the gamma complex, which now acts as a clamp unloader, removing the clamp from the template so it can recycle to the next primer and begin the cycle anew. Let us revise whatever we have seen and studied just now in this lecture with the help of this animation. This animation shows the loading of the beta clamp to the newly primed template primer junction. We can see how the leading strand is continuously being synthesized in the same direction as that of the movement of the replication fork, and the lagging strand is being synthesized in the form of Okazaki fragments, and the lagging strand loops out. The core of the leading slides over the template, whereas the core of the lagging cannot slide over the template. Rather, the lagging strand template is pulled through the core and DNA is synthesized in the form of Okazaki fragments. This continues till the elongation phase is completed. With this, we come to an end of this particular lecture where we have talked about the initiation and elongation of DNA replication. In the next lecture, we will be talking about the termination of prokaryotic DNA replication. If you have any questions, you can ask in the comment section. Thank you.